Welcome again to Let's Talk Forensics with Pastor Dan. We're getting down to the last two concepts. Each of these concepts will be single, all by themselves, one idea, and then we'll just do some practices and stuff like that. The first one we'll look at is the big one that it really exploded in the late 80s, early 90s as far as forensics. DNA. DNA stands for deoxyribonucleic acid. Now, what is DNA specifically? It is the basic blueprints of an individual. DNA is what's really blown up biology. When Darwin first came up with his Origin of Species, he thought the cell was this glob. And we understand today the complexity, the intricacy of DNA. It really blows you away. Literally, it is a blueprint. It is a sequence. From DNA, any part of your body, I can identify every aspect of who you are. Hair color, eye color, size, race, ethnicity. If there's any defects, all of those things are in every cell of your DNA. And it coordinated in such a way that it will turn on and turn off in specific sequences. And if it is so fine to the slightest derivations cause it to fall apart. So DNA is so intricate, there has to be a God. There's no way around it. DNA literally is God's blueprints for us, showing us how intricately, how wonderfully you are made. Now, how is DNA organized? First of all, it's in chromosomes. There's 46 chromosomes, 23 you get from mother, 23 you get from your father. On those chromosomes, we have genes or specific traits. And in those, we have alleles. Now, those alleles are in the sequences that make up 4 of nucleic acids. Those are adenine, guanine, cytosine, and thymine. And they go in a specific ratio. Adenine goes to thymine only, and guanine goes to cytosine. Now, who discovered DNA? When you look it up, most places will identify 2 people, Watson and Crick. Watson and Crick were the scientists who are given credit. They're Nobel Prize winners for DNA. There was a radiologist, a phototechnician, who also helped Watson and Crick. But she died before the Nobel Prize. The Nobel Prize does not give out awards posthumously, so she wasn't awarded, but she also was, her name was Franklin. So Watson, Crick, and Franklin. Okay, who first applied it to forensics? When did it really blow up in forensics? Was a Dr. Alec Jeffreys, Sir Alec Jeffreys, he was English. And the case here that he involved, there was a man in England, in the United Kingdom, who thought he was of noble birth. He thought that his mother was of a royal family. And of course, after the woman passed away, the family didn't want anything to do with him. So he went to Jeffreys and asked them to perform a DNA test on the woman's sample with his sample. And Jeffreys was able to identify that, yes, indeed, he was part of that family, blew up that royal family, and he was able to gain part of his inheritance. So that's the first place that forensics was ever used, and from there, it's just gone off the deep end. How accurate is DNA testing? If you have a DNA sample and it is not corrupted, it is 99.999, basically perfect. You can nail it down to the person, one person and one person alone. Even twins will have slight variation. The only way you can exact would be a clone, and clones don't work because a copy of a copy, we're made in the image of God. And so if we're a copy of a copy, you know what happens to copies of copies? They degrade, they don't get as good. Clone, I don't think it'll ever work. All right, now, when it comes to DNA fingerprinting, this is known as the process of profiling basic sequence of DNA to make a match. And what we're looking at here is the genome, the basic code of the individual. There's what's called the Genome Project, where we're actually going through and identifying all the sequences involved inside of DNA. That's what's really made us understand how incredible they are. Now, there's 2 ways that we can do DNA First is called VNTR, variable number tandem repeats, as a way of sequencing those. And the second one is known as STR, short tandem repeats. Either one of those we can do to get either a longer stream cycle or a shorter cycle in order to get a match in that way. All right, now, where can we get DNA to test? Well, first of all, skin cells. So they'll rub them off the side of your inside your cheek to get those skin cells. Blood contains DNA. It's a fluid, makes it easy. Perspiration, saliva. Often when you're having a case, what they'll do, if they're trying to get a DNA sample discreetly, they'll give a suspect water, and they'll wait for them to put it back down on the table. As soon as they put it back down on the table, the investigators will grab it. Now they have a DNA sample because you released it. You no longer have possession of it. Good attorneys will realize that trick. Either not allow their clients to drink the water, or they'll mix their saliva in there as well so that it's not a sample. All right, hair. Now hair is starred there because remember we talked about before. Hair only works at the cuticle. So if you have to literally pull out, if you cut hair, there's no DNA. It's only at the skin root tag in that way. All right, now, to actually go through the DNA process of DNA fingerprinting, the first step is retrieval. We got to extract the sample that we're going to get. So we'll either swab a blood stain, maybe we'll swab a person's sample, so however we're going to retrieve it. Second, we're going to do extraction. We're going to pull the DNA out of the sample. We're going to do a real simple extraction. We're going to do Gatorade tomorrow. And so the Gatorade itself is the retrieval is the Gatorade. Extraction is using a little bit of soap. And then the third step is restricting the fragments. We do that by adding specific chemicals. Soap, and again, amplification. What you're going to do in the lab that I'm going to show is you amplify by using heat. You're going to put it underneath your armpit to warm it up a little bit. And then lastly, electrophoresis. We're going to do a different process instead of electrophoresis. Electrophoresis is the actual process that you go through to do a DNA sample. We're going to just add ethanol. Ethanol will pull the amplified DNA samples out of the soap and be able to identify and see it in that way. All right, now, what is the process of DNA? Now, when we do the process itself, we're going to get something that looks like this. So we're going to get a pattern of separation. From that, we can match up directly and be able to see how it works. Now, how does the process work? First thing you can do is you can take agar, like a jelly, and put on the sample. You're going to load samples, the same samples on both sides, and then you're going to electrify it. As it electrifies, it's going to pull it across. It takes a long time. It takes 20 to 24 hours to pull that across. Once you get it separated up, then you get this sequence you see over here, right there. And then that is how we are able to see the DNA and make a match in that way. All right, now, what can DNA prove? First of all, can prove the presence at a time. You were there at some time. Doesn't prove that you did it, but that you were there. Second, paternity, maternity, like we say with Al Jeffries. Mama and daddy. Who's your mama? Who's your daddy? Those kinds of things. And that's dead on because you got your DNA from your mother and they can match up in that way. Brothers lines. Identify remains. And of course, the biggest situation for this was 9/11. There are still remains that are so fragmented, so corrupted that it's hard. But the DNA process is improving. So every time it does, they take more of that samples out and see if they can find more and identify it so that the next of kin can know that the remains were found. All right. Now, a system we have is called CODIS. CODIS is called Combined DNA Index System. It's like IAFIS. It's like IBIS, integrated automated fingerprint identification, integrated ballistic identification system. CODIS is for all DNA, and it's DNA sample obtained voluntarily or through court mandated for who gives DNA voluntarily? Soldiers. So their remains that they should be killed can be identified. anybody that's in the service of any type. And if you're convicted of a crime, specifically a sex crime, your DNA will often be mandatory retrieved and is kept by the FBI. All right, now, the crime of the century. I want to talk about this one because this is a situation where it should have been a slam dunk. What I'm talking about here is Orenthal James Simpson, otherwise known as O.J. Simpson. Now, the case. Nicole Brown Simpson and Ron Goldman were dating, and their bodies were found in Brentwood. O.J. Simpson, Nicole Brown was his ex-wife. He was known to have a bit of a temper, and her DNA blood sample was found inside of his Bronco. His DNA was found at the home. Now, that's the evidence. It should have been a slam dunk. But there was definitely poor procedure. People had come through that crime scene, so that brought a lot of contamination, a lot of photographs, things along those lines. And there's also a bit of a bias. You see, Los Angeles is the second largest city in the United States, but it does not have enough of a police force to properly secure the city. In reality, they basically contain more than and as a result, there's a lot of distrust of the police. And so that's the reason why it was really easy for the jury to see the police as this biased group who was trying to frame O.J. Simpson. When they did the case, there's two possible reasons. One, they actually did it wrong, or two, it was just bad procedure. Two main things that I think that the defense did wrong, the prosecution did wrong. First of all, what's his name? Ron Furman was a lead investigator. And Ron Furman had been known to say a certain word to people. Starts with an N, you know what I'm talking about. As a result, they made him say that word over and over and over again in court. And it made the jury see him in a not very good light. Second was the glove. If you ever remember the photograph of when they made O.J. Simpson put the glove on, well, first of all, it's a leather glove. Leather restricts when it's not worn for a while. Second, O.J. Simpson was an actor, so he could play the part. And third, his defense team had him stop taking his arthritis pills. So his hands swelled up. As a result, it didn't fit. Based on the evidence I saw, do I think O.J. Simpson did it? Yes, I'm sorry. But according to our double jeopardy system, he was acquitted. He was found not guilty. He was eventually found liable in a civil case and basically took any proceeds he would have ever had away. So that's the case, essentially one in which DNA should have been a solid, but because of the fact that there were a lot of factors in there that were really, really bad, it messed it up. All right, that's our DNA. That's what we talked about. Any questions, let me know. Take care. God bless. Bye.